The overall goal of this procedure is to isolate chromatin-bound proteins from the mouse heart. This is accomplished by first homogenizing the heart and then isolating cardiac nuclei via a sucrose density gradient. Next, the nucleoplasm is separated from the chromatin fraction. Then, proteins are extracted from the chromatin pellet to enrich for proteins loosely bound to the DNA. Finally, proteins are extracted from a different chromatin pellet to enrich for proteins tightly bound to the DNA. Ultimately, results can be obtained that show the characteristics of the nuclear proteome under different physiological conditions through quantitative label-free mass spectrometric analysis.
This method can help answer key questions in the cardiac field, such as how chromatin remodeling contributes to cardiac hypertrophy and failure.
After sacrificing an adult mouse and excising the heart, rinse it in ice-cold PBS, then place it in a glass ounce with 2 ml of lysis buffer containing a protease and phosphatase inhibitor mixture, and homogenize it. Pour the lysates through a 100 micron strainer and collect the filtrate in a 1.5 ml tube. From this point forward, unless otherwise specified, keep the sample on ice. Centrifuge the sample at 4000 rpm for 5 minutes at 4 degrees Celsius. Collect the supernatant containing the cytosol and store it at -80 degrees Celsius. Resuspend the pellet in 200 microliters of lysis buffer. Add 1 milliliter of sucrose buffer to a new tube and gently layer the resuspended pellet on top of the sucrose.